introduce Michelle Schneider-Berry. She's the director of the Herbert and Jacqueline Krieger Klein Alzheimer Research Institute at Rutgers University. She has her undergraduate and graduate training in Israel, and she came to the United States and became an assistant professor in Mount Sinai School of Medicine in 2001 and rose through her ranks to become full professor in 2023 before she came and joined at Rutgers. She's considered as a leading authority in metabolic derangements in brain aging, cognitive decline, Alzheimer's disease, and dementia. Her research spans both epidemiological study of dementia to clinical trials, which test novel treatment pathways for prevention of the disease. She has won several professional awards, has 220 peer-reviewed articles, and has been continuously funded through NIH, Alzheimer's Association, and other research foundation. And today she's going to really share light to us so that we can partake in understanding how these two organs potentially play similar roles and with similar risk factors in sight, and how we could be interdependent in our research as we craft our therapeutic journey for our patients. With that, Michelle, the floor is yours.
Thank you so much. I hope you don't hear all the kids in this background noise. I'm in a place that I was sure will be extremely quiet, except that it isn't. Partha said before that it's not so noisy on your side, so I hope that's the case. I'm going to push a lot of data on you, so I hope I make it interesting. And because I thought I have an hour and 15, but I have only an hour, so there's going to be a section that I'm going to run through the slides without going one by one and just summarizing it for you. But hopefully this is going to be interesting and relevant to your research and clinical life. So some basics for us to kind of speak the same language, okay? Diabetes is a risk factor for dementia. And what I mean by that, both vascular dementia, which today is essentially a disappearing name, and of course, Alzheimer's disease. And soon I'm going to possibly contradict myself a little bit, but diabetes type 2, okay, I'm talking all the type 2 diabetes is a risk factor for dementia. But it overall about doubles the risk of developing dementia. The same for MCI. MCI stands for mild cognitive impairment, which is this period before dementia is ascertained that there is some cognitive impairment, but the function is still okay. And diabetes is also related to a faster rate of cognitive decline. Now, naturally, we want to understand what are the underlying mechanisms or underlying pathological processes that explain this association between diabetes and cognitive outcomes. And there are studies out there showing a little bit of everything. Okay, that diabetic patients have more cortical thinning, they have smaller hippocampal and temporal lobe volumes. But the bottom line, the most consistent findings are that diabetics have more cerebrovascular disease, they have more strokes, like coronary infarcts, microbleeds, white matter hyperintensities, reduced cerebral blood flow, and reduced cerebrovascular reactivity. Now, what about Alzheimer's neuropathology? Because I showed you just the epidemiological evidence suggesting that diabetics have more Alzheimer's disease. And the reality is that that is not correct when we look at the pathology. So both old studies and a little newer studies all show that diabetes is not related to the two hallmark pathologies of Alzheimer's disease, which are neuritic plaques, which are based on aggregates of amyloid, and neurofibrillary tangles, which are based on aggregates of the tau protein. And that, at least on those post-mortem neuropathological studies, what is explaining the relationship between diabetes and dementia is cerebrovascular disease. I'm going to introduce you to a study that is ongoing in Israel, because it was the basis for much of our understanding, at least in our group, of how metabolic impairments in general contribute to brain aging and cognitive decline. So the cool piece about this study, it's a study of about 1,000 type 2 diabetic older adults. They're above 65 years old. We have been following them for the last 10 to 12 years. And what is, and as you see, I have still the Icahn School of Medicine, Mount Sinai, because this study was funded for a series of times while I was there. And it's a collaboration with the Sheba Medical Center in Israel and the Maccabi Health Services. So I'll take a minute just to explain what this Maccabi is. Israel has socialized medicine, and it has four HMOs, four, so much noise, I hope you guys are hearing this. So there are four HMOs in Israel. The second largest is the Maccabi Health Services. This is the MHS here. And we have the data for those 1,000 participants since 1998. Now, everybody goes there. Rich people, poor people, smart people, stupid people, everybody receives services from them. So it's a really nice representation of the country. Now, this is just to get a feel of what happens when you have really, really good data sets from an HMO like the one that we have, right? So those are our participants, and those dots are their measurements in those over 12 years for cholesterol, irrespective of cognition, okay? And what we show is that over time, their cholesterol goes down, and this is highly likely because they receive statins. And this is the A1C for our participants. So first you see that they have many more dots, and this is because cholesterol is typically measured once a year in one's annual, while A1C is measured 3, 4, 5 times a year. And you can see that no matter how well and how aggressively diabetes is treated, there is a slight tendency of A1C over time to go. So we asked the question, the favor of this opportunity to look at really long-term trajectories of hemoglobin A1C, and can we check or can we examine if it is associated with cognitive impairment? And what you see in this graph are actually empirical group trajectories of hemoglobin A1C over time. And what I mean by that is that we didn't give the computer any assumptions. We asked the computer to tell us, based on the data you have, tell me groups of people whose trajectories of A1C seem to be similar. And what you see is 3 trajectories at the bottom that are mostly okay, right? They're relatively stable, and they are going up very, very slowly. And the 3 upper lines, right, in this graph, they're actually much more noisy. There's smaller numbers here as well, but those are more severe diabetics. They have higher A1C, and the red and the upper red one, clearly the doctors are making every effort to get those people to a good A1C, but over the lack of success, either because the patients are not adhering to their medications or because they have very, very severe disease. But what we found here in relation to cognition is that each one of those trajectories is related to cognition in an incremental way, okay? So the people who are diabetics, but have a nice, well-controlled, reasonable A1C, they have very little cognitive impairment. While those people up here, and the black line as well, that have glucose dysregulation, clearly their glucose control is not good, their cognition is far more impaired. Yeah, so in this context, we started asking the question, and especially because of those studies with negative associations with Alzheimer's and pathology, we said, okay, so maybe the drugs for diabetes, if they stabilize glycemic control, or through some other mechanisms, maybe they have something neuroprotective. So this is data from Israel, but it's very similar to what the CDC has in the United States, by the way, that at least in the beginning of the course of diabetes, about 90% of the people are immediately treated with diabetes medications, and about 10% of them take a longer time until they're treated. The doctors try to give them a chance to control it via a lifestyle. And eventually, most, if not all diabetics, as you know, are treated. And when I was at Sinai, I was fortunate to work very closely with Harry Haratunian, who runs the brain bank there. Sinai has over 3,000 brains of people who donated the brain for science, the majority of them participating in aging studies. So we were able to match postmortem brains from people with and without diabetes and see what's happening in their neuropathology. And don't worry about this. I'm going to walk you through, even though it's a little heavy, but I just want you to, for those who are not familiar, this is an neuritic plaque. This is an amyloid plaque. This real brains from the sine and brain. And this is a neurofibrillary tangle. Those are as I mentioned, the two hallmark pathologies of Alzheimer's disease. What you see here, to make a complex story simpler, is that people who, before death, were diabetics and received either, insulin and another diabetes medication, they seem to have far less neuritic plaques, amyloid plaques in the cortex, hippocampus, as well as in the amygdala. For a density of neurofibrillary tangles, you can see that this, by the way, this effect, as big as it looks, is not significant as trend level, but you can see a direction in the cortex, but not in hippocampus and amygdala, and we can discuss this later if you have questions about it. But what's important and interesting here is that in contrast to no diabetes or to diabetes with one of the drugs, right, either insulin or another diabetes medications, those who at some point had a combination of those seem to have developed less pathology. And if you're asking yourself, remember, those are brains that were donated, and this paper was published quite a few years ago, so there were no GLP-1s on this, okay? We'll get to GLP-1s, but those are primarily sulfonylureas. So we asked ourselves what those results may imply. Okay, now we have to remember that if any post-mortem human study is only observational, right? Because we can't do longitudinal studies on longitudinal data, post-mortem data, sorry. And still we can ask questions of what those results might imply from a biological point of view. And we took the direction of investigating the insulin receptor signaling pathway. So I'm just walking you briefly through two studies that supported our postmortem pathological studies, but in a dish. So this study actually looked at metformin in a dish, and metformin insulin, you know, that it degrades amyloid, okay? But metformin happens that in this study was actually leading to further aggregation of A beta, counterintuitive, but that was the case. However, in combined use, metformin enhanced the effect of insulin in reducing A beta levels, suggesting again, it's something in this game between insulin and other medication might be hitting different areas of different spots in the insulin signaling pathway, leading to neuroprotection. And this is also a basic science study showing, again, that A beta oligomers, which was kind of the precursors of the amyloid plaque, they sit on the insulin signaling and downregulate the whole pathway. However, if you put insulin, that downregulation is partly reversed. And if you add to it rosiglitazone, which some of the doctors here might not even know what that is, but was a diabetes medication that eventually was so black box that it had to go out. But when you put both of them, then that downregulation by A beta oligomers is essentially fully reversed. So the insulin signaling pathway works essentially normally. So this made us think a lot about this. Why is this combination I wish I might somewhere else to go than there. So I hope you guys can hear me. In any case, we moved on and we did a study in animal models. So those are Alzheimer's animal models that were treated either with saline as placebo, a GLP-1 exenatide at that time we did, intranasal insulin, and the GLP-1 the examiner was chosen because there was a prior infection that if you splash it into the nose, it goes into the blood-brain barrier, through the blood-brain barrier for those animals. And of course, a combination of both. And what we showed there, as this title says, that those animals on the combination therapy, they had both better memory than the other groups, but also they had a normalization of the expression of the insulin receptors pathway genes compared to control animals. And finally, we have a study, so the original study that I mentioned looked at the pathology. We looked at homogeneity. Okay, so you take a little bit of the brain, a piece of the brain, you mush it, and you look how much pathology there is in there. In this case, we actually assumed or hypothesized that most of this medication's effect must be happening on the vessel. So we isolated vessels from those human brains, and we showed that indeed most of the effect or most of those differences that we see between type 2 diabetics who are treated or not treated, those people who do not have type 2 diabetes are actually occurring in the vessel. So I said, I mean, if you notice, but the title of my talk included the expression from bed to bench to bed. And this is one example. So the first bed is that the observation that diabetes seem to be related to cognition or to cognitive impairment, and that the drugs might have a protective effect. And the bench is the animal model study that we ran. And the back to bed is a clinical trial that is now funded by the Alzheimer's Association Public Grant. So I don't have data to show. I'm just letting you know that this is happening. And we are running a trial. It's a 2x2 trial. So there are people who are receiving post-saline in the nose and a pill that is placebo. And people are receiving intranasal insulin. And we have a third group of semaglutide, GLP-1, and a fourth group that receives both. The study is ongoing. We chose to work with people with mild cognitive impairment and with metabolic syndrome. And the reason for the mild cognitive impairment is because it increases the chance of having an effect and more cognitive decline because they're already impaired. And metabolic syndrome is because of our postmortem study where we show that there seems to be a particularly strong effect of this combination on the blood vessels, and people with metabolic syndrome have more cerebrovascular disease. So I'm going to close that piece, okay? And maybe a year or two, if I come in and give another talk, I have results from this clinical trial that I can't tell enough how eager we are to know what the results are going to be. Now, because Partho asked me to shorten a little bit the talk, this topic of the role of advanced glycation and products in cognition, I'm going to really rush it. And then at the end, I'll leave enough time for questions. And if you want, we can go back to. So one thing I want to say that every time I say AGEs, it's not age, it's not chronological age, it's advanced glycation and products. So just stay with me on this. So AGEs are non-enzymatic reactions of amino groups and proteins and also lipids, and with reducing sugars. Okay. Sometimes people think that ANC is an AGE. It isn't. AGEs are a long chemical cascade. And it has numerous types. I put this old slide purposely, okay? And the reason is that the measuring AGEs is not easy. The AGEs that are mostly examined, we're using primarily ELISA. Some are measuring with more precise measures, so let's put it that way. Methylglyoxal, CML, carboxymethyl and pentosidine are the three most well-measured AGEs. And still, there are many differences between labs, which makes it a problem. And the reason why I'm saying this is that we're developing advanced mass spec techniques that hopefully will shed better light on this. Partha, you want to say something?
Yes, so your voice, I'm just wondering whether the microphone is breaking up a little bit.
Okay, so I'll try to hold it next to your nose. Is that better?
That's better, yeah.
Okay, very good. So the two main pathways via which AGEs affect health in general is through the receptor for AGEs, which I'm sure you know, the RAGE receptor, right? Leading to pro-inflammation or to inflammation and to oxidative stress. And the second pathway is through protein cross-linking. And there is sufficient literature out there suggesting that AGEs cross-link amyloid and in fact preventing amyloid from degradation. Dietary, so AGEs are primarily endogenously generated, but they're also present in the diet. Okay. In diabetics, because they link to glucose, diabetics tend to have high levels of AGEs, but foods also have high levels of AGEs. And that, just to make it quick and short for you, all the yummy things have more AGEs than the less yummy things. So protein, okay, foods rich in proteins such as red meat and very high temperature or high temperature have generates more AGEs than foods that are raw or fruits and veggies. So this is just a slide to give you a sense. This is my friend and colleague Jaime Ribari from Sinai that had the guts, really the guts, to develop a kind of a diary of 750 foods on which he did ELISAs and showed the differences in different types of cooking. And so you get a feel in when chicken is boiled in 1,000 units of the AGEs. Well, if it's boiled, which is yummy, it's 6 times higher. Now, in mice, okay, I emphasize. Partha, can you hear me good now?
Yeah, you're good.
Better now that I'm holding? Okay. So in mice, there were several studies feeding animals with high AGE diets, which essentially means that their pellets are cooked in high temperature. And those studies, numerous studies show that those animals develop more type 1, type 2 diabetes, more atherosclerosis, kidney disease, insulin resistance. They have a harder time healing diabetic wounds. There's a beautiful study also on short lifespan and higher mortality in animals that are fed with high AGE diet. And we asked the question about dementia. So now I'm going to rush it a little bit, but overall, we show that both in blood and in the gut, if they're higher in the blood, if they're higher levels of AGEs, and in the gut, if people report higher AGEs intake using a specific food frequency questionnaire that is adapted for AGEs. Okay, so many questions. There are no questions on veggies and fruits because they barely have any AGEs, but many questions about how you cook your meat, for example. So all our studies essentially show that those who are either eating or have, for whatever reason, high AGEs levels, for example, because of diabetes in their blood, their cognitive decline is faster. We also did a couple of animal model studies in which we show that animals fed with high AGE diets, they just don't learn. So those are the Alzheimer's animals doing a task of the North water maze that usually they very quickly discover where a little platform is, so they didn't have to be floating in the water. And the animals were sitting high AGE diets, simply cleaning there. And another study, this is a study on different animals that we had, but what this study was able to do, and our first study wasn't, is to measure AGEs in blood and in brain of those animals and show, again, animals, that there is a very nice correlation between the levels of AGEs in the blood and in the brain. So with my colleagues at Sinai, we received a set of brains from diabetics, non-diabetics, with Alzheimer's disease, without Alzheimer's disease, and who had both human brain tissue and blood. And what you see here, the right side of this slide is a brain. So what you see here is that brain carboxymethyl lysine is very nicely correlated with carboxymethyl lysine. If anyone is concerned about this potential outlier, when it's out, the change in the correlation is minimal. And the same for brain methylglyoxal is the last very nice correlation, which to me is mind blowing, right? That the brain is theoretically so affected by what wheat. Yeah, this slide is just to tell you that people with both diabetes and Alzheimer's disease were the ones with the high levels of blood and brain AGEs, and people without those two conditions had the lowest levels. We also, in the original study that I showed you, I passed it through it very quickly, but it was a small study on dietary AGEs and cognitive decline. So we got somebody from the NIH to ask this question in a much larger sample. So here, these results are on over 650 people in the Rush study in Rush University. And once again, we show that reported higher dietary intake of AGEs is associated with a faster rate of cognitive decline. So he comes up from bed to bench to bed again. And my really incredible student, who is now Dr. Goni Latani, she ran a clinical trial, which I'll tell you the bottom line right away because I want to have time to go into other topics. But the bottom line is that doing a low, in this case, right, it's in humans, so you do a low AGE diet. A low AGE diet is feasible in older adults. So people, it was a 6-month study. People were able to handle the diet, and the majority were relatively highly adherent. The second takeaway from this clinical trial is that people on the low AGE diet versus a typical American Diabetes Association diet, which was a control group, their AGE levels went down over time. So a diet, intervening on the diet with a low AGE diet does affect your systemic AGE level. Yeah, but of course, what we most wanted is to see a change in cognition, which we did not see. Okay, and now I want to take this a little bit with myself. The first thing is that at baseline, cognition was associated with AGEs. Now that baseline likely reflects the lifetime intake or AGEs levels of that person, and it is correlated with cognition, as similarly to what I have shown in the other studies. The issue that I think happened, but I'm not sure, it might be a zilch. It might be that dietary AGEs don't do anything to cognition. But if you look at the clinical trials today that are running dietary studies, they treat people for at least 2 years. There are clinical trials that are taking 5 years doing the dietary intervention. So those 6 months might have been too little. And the other issue is that we did the mistake of mixing. We didn't want to take people with dementia because that would mean that all the time we depend on something else and making sure that the reporting of the diet or the intake was correct. So we took people both with normal cognition and mild cognitive impairment, and we have some signal in favor of a low dietary AGE on the MCIs and people with already some cognitive impairment. We have no signal whatsoever in those normal cognition, suggesting that that might have been a design mistake. Now, remember that I showed you that old slide, and I said I'm purposely showing you an old slide of how we measure AGEs with ELISA. So based on those many results, I've been really fortunate to engage in a collaboration already. 4 years, we're working very closely together. With this group of people, Yishai, David, and Hila are from the Weizmann Institute, and Itzik is from the Shiba Medical Center. And Nini is a dentist that got fed up of being a dentist and did a PhD in bioinformatics. And what we did together, my Weizmann colleagues, of course, were the ones leading this effort, was to develop a glycoproteomic method that would measure not AGEs per se, because AGEs are a more advanced stage from a chemical point of view, but would measure both glycation and glycosylation. And now I'm going to walk you through the way that it made me understand this. For some of you, this might be trivial. For me, it wasn't trivial at all. Okay, so pardon my simplicity, but it does help, I believe, understand what is that we're accumulation. Okay, so what is a glycopeptiform? Okay, glycopeptiform is the word that I'm going to be saying many times for 10 minutes or so. So this is a peptide, okay, and those are glycans, and the glycans sit on different sites of the peptide and define much of that peptide's function or that protein function. In proteomics, what does the mass spec do? It takes away all the post-translational modifications, really clears all the post-translational modifications, and you're left only with the protein. In glycomics, the mass spec cuts the glycan, but it takes away the protein and all other post-translational modifications. What are we doing with our glycoproteomics technology? We actually cut the peptide with the glycan. So we know what the protein is, we know what the glycan is, and where is it sitting. What we miss here is the structure of the glycan. Okay, we can't see 3D. We know what are the different components of the glycan, but we can't see it 3D. But still, we get the glycan, its components, the sites where it's sitting, and on what protein. This technology is able to look to assess and measure both glycation, which we have talked enough about, and glycosylation, which is, as I said before, glycation is a non-enzymatic reaction, is a much less exciting reaction than glycosylation, in my opinion, simply because glycosylation is an essential feature of the cell function, including the lung. But also because glycosylation is the most common post-translational modification in the body. And now I'll show you a few studies that we are doing. Some of them are ongoing, some of them we are just finishing. And so this is all kind of hot potatoes, right? This is our very new data. And so in one study we took, and it's a small study of 22 people. Let me show you this. So those are 22 participants of that IBCD study that I showed you before. For me, we had 2 times of blood, so we had glycoproteomics measured twice, and we had cognition at 2 times this. Half of them were strong non-converters. Okay, there were people that in their 36 months with cognition remained abundant with perfectly fit. The others had the same level of ANC, education, more or less a match for sex, baseline cognition, etc. at baseline. But then over time, they decline and they either converted to mild cognition or to dementia. So notice that with this novel technique, in blood, because soon I'm going to show in brain. In blood, we were able to identify about 3,000 glycopeptiforms from about 300 proteins. What those 2 figures show is just the unbelievably enormous variety of those glycopeptiforms. Okay, so you can have many glycans on one single protein. Okay, you can get to over 100 glycans on one single protein, and you can have also many glycans on a particular site. Okay, so I would think in the beginning, one site, one glycan, and that is not the case. There are certain sites that have many glycans. So essentially, from my point of view, we're developing a field of research because we know very little about this. So what we showed in this small study, okay, that in older adults with type 2 diabetes, the expression of several glycopeptiforms differ between decliners and non-decliners, both at baseline and over time. Now, why this is important? Because I told you, right? I told you before that those patients were very well matched at baseline. They didn't differ on both metabolic and cognitive measures. And nevertheless, their glycoproteomic profile had some glycopeptiforms that were already different at baseline and further changed more significantly, and I'll show you an example in a moment, at follow-up 36 months later. Now, this is, from my point of view, we're in the beginning, okay? So, I mean, we have to be very careful with what I'm saying right now. This is an example. I can say this is a SAMP, a protein that you can see that in non-decliners, the glycopeptiform expression level declined, declined some over a period of 36 months, but in the decliners, that decline was far more dramatic. The reason why this is important and the differences at baseline were already beginning to show is that you guys as doctors, the whole issue of biomarkers is a little bit of a problem, right? Because the biomarker is supposed to walk with the phenotype, in this case, cognition. But you as a doctor, you see a person, you want that time that you see that person to have a blood draw and to say, oops, there is a problem. This person has a very low number in this glycopeptiform, which is highly suggestive of cognitive decline. So there is a set, and again, we have to replicate it, and we're replicating this in 250 people right now in blood. So we have to be careful with what I'm saying now, but this is very exciting and maybe beginning to suggest a type 2 diabetes related signature for cognitive impairment. Maybe I'll skip this, but just for anyone who's been interested, not surprisingly, the glycated peptiforms, the pathways that were more predominant in a genontology analysis were metabolic pathways, and for the glycosylation, it was primarily immune response pathways, both of which are well known to be implicated in Alzheimer's disease. So I emphasized many times that what I was showing you up to now was blood data, and now I'm showing you brain data, okay, which is also very exciting. So this is a study on 366 participants for whom we had what you see here, a DLPFC is dorsolateral prefrontal cortex. And some people sometimes ask, why didn't you get hippocampus, which is the first region affected by Alzheimer's disease? And the reason is that nobody shares hippocampus because it's so tiny. But the dorsolateral prefrontal cortex is implicated in Alzheimer's disease from so many directions that we feel very comfortable about using it. Remember that before I showed you that in blood, we got about 3,000 glycopeptiforms, in brain, we get over 11,000. And before I showed you that it was from about 300 proteins, and in brain, we're getting over 1,300, and with time, we're getting better. Now, this is a complicated slide. I'm going to break it down for you, because each piece of it, I think, is meaningful and opens new directions for research. So the blues here are showing that upregulated glycopeptiform is related to protection, to less cognitive decline. The pink ones show that upregulated glycopeptiform is actually neurotoxic, is related to more cognitive decline. Now notice that there is no protective glycation. All but one of the pinks are glycation. Glycation is neurotoxic. Glycosylation, in contrast, some are protected and less so, but some are toxic, neurotoxic. Notice also, and this is, I'm personally very excited about it, but of the 5 glycopeptiforms that withstood Pumfaroni adjustment for 11,000 comparisons, 3 of them are neuronal contractin. Okay, so you have neuronal contractin A, B, and C. Now, neuronal contractin is synapse protein, synaptic protein, and I'll tell you a little bit about things that we're trying to move on and do about this. Another one that is interesting here is the amyloid beta precursor protein. If it is glycated, it is associated with a faster rate of cognitive decline. And the last thing that is also blowing our minds in this slide is this glycan composition. So if you remember when I show you the little snake, the little peptide with the glycans have different compositions, right? So those little pieces are those interesting letters and numbers. And this glycan composition, that is about a third of the ones that were significantly associated with cognitive decline. When you look at the full 11,000 glycopeptiforms, they make up less than 0.02 of those of the whole glycopeptiform profile. So this tells us that this glycopeptiform composition, for some reason that we don't know yet what it is, has to do with cognitive decline. And I have to tell you that we search every possible data sets and databases available in the market to see if anybody knows anything about this glycopeptiform, and we found nothing. So it's something we're going to have to address. Yeah, this is just a different way of showing the same thing. So I'm going to skip. And here is just to give you a more clinical feel of what we're saying. Okay, so glycosylated neuronal contractions. If you look at a person that has the lowest levels of this protective glycopeptiforms, they have a fast rate of cognitive decline. But if the person has a high level, let's say in the 90th percentile of the neuronal contraction, their cognitive decline is far slower. So this is, I think, from therapeutic point of view, an exciting direction to take. The other thing that is important here, how am I doing with time? Sorry. I'm okay. That's good.
5 minutes. Yeah.
5 more minutes is good. 5 more minutes. Okay. So for me as a clinical neuroscientist, I always need to understand where's the therapy at the end of the tunnel, right? So I want to explain to you or give you an example of something that is from our data that gives a sense of how we can further investigate this stuff. Okay. So APP, the site of the glycated APP that I mentioned before, APP is an amyloid precursor protein, is actually sitting, the glycan sits on alpha secretase, okay? On alpha secretase. Now, alpha secretase is what cleaves the amyloid protein, right? The A beta domain of the amyloid protein. So if that site is taken by a glycan, then beta secretase and gamma secretase are free to essentially generate and produce more A beta. So this is just a little example of how understanding where those glycans are sitting and how they're changing the function of the protein might be affecting a cognitive decline. And this is very, very brand new. So I'm really bringing you very new data. This is my dear colleague, Yasser Ituria Medina from McGill University. And he's although he's an engineer, but he's de facto a bioinformatician. And he developed this very, very cool technique, which he calls a mathematical technique, which he calls contrastive trajectory analysis. And simplistically speaking, it maps how omics, and he uses every omics that are available for a particular participant. So he maps how omic profiles change from normal aging to dementia. And in that way, he kind of identifies distinct molecular pathways of disease progression. So we used those brains, that the 366 brains, I sent them to him, and he is blind to who have and who doesn't have diabetes, but he ran the glycoproteomics, and he ran them on this contrastive trajectory analysis and found 3 such trajectories. So those are profiles of glycoproteomics that are built of different types of glycopeptiforms. And we see that there is one that is related to diabetes. Okay, so one subtype of glycoproteomics seems to be suggesting a molecular signature that is specific to diabetes. And we are working very hard on this, but this gives you a sense, again, of how we're trying to disentangle the role of diabetes in the brain from a molecular point of view. So just to conclude, there are subgroups within diabetics. Not all diabetics become demented. And this is important from a clinical point of view, from a clinical trials point of view. We need to continue trying to identify who are those whose brains are most vulnerable to dementia. As I showed you, the combination of T2D medications, right, the type 2 diabetes medications, possible because they address a range of brain pathologies or different areas in the insulin signaling pathways, they might ultimately be serving to help cognitive. And I'm going to skip the conclusions about injuries because I did much of it while we were talking, but I do want to just emphasize that if I had done that clinical trial today, I would not measure injuries in simplistic ways. I would do the glycoproteomics and get really the full picture and would work with only MCI adults and not with with normal control. And just for ongoing and future steps, anyone who wants to join us or is interested in this area, of course, you're invited. So we just got a grant to begin to do glycoproteomics on 800 brains. With neuronal contraction, we're now beginning to do targeted glycoproteomics to see if our finding is true, both in CSF, and we're also isolating synaptic cells, because it's a synaptic protein within your seat, it's happening there. And of course, we are increasing dramatically our glycoproteomics in blood studies because from thinking point of view, that's going to be much more forward. And I need one more minute, Martha. I just want to push for a morning. In Rutgers, we're building now. We just finished recruiting the first South Asian middle-aged high-risk individuals to participate in our longitudinal study of cognitive decline and dementia in the South Asian population. The reason why we want to focus on them is because there is barely any research on them, and definitely not still research. And it's it's a population with the highest immigration to the United States right now. There's 5.5 million people in the US. We know nothing, but also because the South Asian population has this incredible feature, which I do not understand, and I find it fascinating from a public health point of view, where they seem to have a lower risk of developing dementia unless they have diabetes and other cardiovascular risk factors. So compared to whites, blacks, Filipinos, Japanese, Chinese, South Asians have lower risk for dementia. But if they have diabetes, they have the highest risk. So I put this on for you to please refer people to us, to ask us questions. It has been a challenge to recruit for this study, which makes me even more excited about doing it. And those people are deeply characterized. We're working, as Partha mentioned before, with Kabir, with a brain imaging with David Zaub. We're working very closely to image their brains in depth. And we're measuring everything. We're measuring blood biomarkers, brain imaging, as I said, of course, cognition. We're doing some digital health, very cool digital health on them, lifestyle, etc. A lot of social determinants of health. The emails that you see here, this is me. Karen and Akanshi are the research coordinators, and this is our center's email. If you have people that are between the ages of 45 and 70 and want to participate in longitudinal research for cognitive decline, please do refer them to us because we're very eager for this study to be successful. And just a big thank you to my Weizmann Institute colleagues, my Russian colleagues, I work with them very closely. At Rutgers, our little team that is slowly growing, and Becky, who has been instrumental for existence. At Mount Sinai, Mary and Harry, and at Shiva, my friend and colleague, she runs the show. So I'm going to stop sharing and repeat to the questions.
Thank you very much, Mikhail, for that very extensive review. I have to admit that I try to understand as many of those pathways, and I find it fascinating of how much little as cardiologists we think about some of these pathways that could be interlinked. But let me start pointing to you certain areas where we have definitely need some help. First of all, let me ask you a question. You mentioned about some amyloid related proteins and things like that. How far is this diabetes related advanced glycation products that you mentioned and this association with amyloid microangiopathies in the cerebral region? Is there a relationship? And if there is a relationship, what is the propensity of bleeding? Because we see a big problem in some of these senile people above the age of 75. We do them PCIs with percutaneous pulmonary interventions, and we give them this antiplatelets and they bleed. And then we find that they have amyloid angiopathies, and many times it's this constellation of diabetes and amyloid. Could you just share us? And I'm thinking if your proteomic process could identify someone who's at higher risk of having a bleed, that is something that we've not been able to predict at the moment.
So, yeah, I'm going to say that the data that we have now cannot ask that question yet. Okay. However, in the SAMART study, in this study of the South Asian community that we're doing, we're collecting a lot of vascular data. So we're collecting cerebral blood flow and blood-brain barrier leakage and white matter hyperintensity and cerebrovascular reactivity, which would get us a little closer. And of course, we're collecting bloods to do glycoproteomics. It would tell us a little bit more on how well they are related. At this moment, all I have is they're related to cognitive decline, and they're related both in blood and brain. Okay, this is essentially as far as we go. And I do want to say that because we found these findings of those, what I call biomarkers, but we have a whole long way before we can truly call them biomarkers in diabetics that decline cognitively over time. And because we know that most of the pathology underlying a cognitive decline dementia in diabetics is from vascular origin, then I think your question is very much to the point, and that a year or two from now, we're going to know to answer it at least better.
And I think we should be very happy to collaborate because we have a lot of physician champions here who are interventionalists who constantly struggle with this whole issue of who is at risk of bleeding with antiplatelets and all. And that's been a big problem for us.
So let me mention something about this that is not directly related to my presentation, but I didn't say a pitch from them. The new medications for Alzheimer's disease that have been approved by the FDA recently, this issue of bleeding is a neurodegeneration. Okay, so they, because I mean, simplistically speaking, when you give those monoclonal antibodies, the amyloid plaque is very sticky and it sits on the vessel, and then you break it, so you harm the vessel. So there is both inflammation and bleeding, and those are the most dangerous adverse events, for those medications, especially people with antiplatelets.
And what about this latest paper in Nature on lithium deficiency and Alzheimer's? It was like characterized, it was all across the press. And I was
the press went crazy. And I think it's a very, very interesting new direction to take. And the main issue, and the press just as much as I do, is that people in the press have absolutely make no differentiation between causation and association.
Yes.
And the study is interesting. They replicated results in different samples, etc. I mean, the study is very well done, no question mark. And I think the direction is interesting. And there are two big problems. One, the first one is that we have cured gazillions and gazillions of mice and animals from Alzheimer's disease. And until this is done in a true serious clinical trials, we won't know if it works. And the second thing is that I think it's going to be complicated to do a clinical trial, or at least a big clinical trial for this, which would be what it truly deserves, because there is no patent there. The drug companies are not going to put it down.
And then I think Dr. I don't know if Dr. Bukhari is around, maybe he's around. So, in the area of rare diseases where the brain and the heart are very connected, I think we are extremely interested on newer molecules and newer pharmacotherapies that may be impacting. And Sabi, you wanted to just, if you can unmic yourself and you can comment about your interest about the potentials of brain imaging and heart imaging. Any other questions related to that?
It was an excellent talk. I enjoyed your talk, and very interested in also amyloid in the brain, because a lot we don't know about. also we have lots of amyloid patients, as you know, that with TTR amyloidosis, they are also at a higher risk of brain bleeds, and they get subdural hematomas. They also get coronary amyloid angiopathy. So I think that's something which we really would love to work with you and do some more work on that. Because as you know, the TTR, 5% of that is produced by choroid plexus and is deposited in the brain and the small vessels, and makes them more prone to bleeding.
So for curiosity, does anybody know if the Salvation community has new amyloidogenesis? Not the brain, heart.
Right. So that's a very good question. So I think Navina is also online. We are just starting a study in South Asians looking for amyloid in those patients. I just reviewed a paper which was from AIMS in India, in which they looked at amyloid in patients who had SAVR, who had surgically aortic valve replaced. And there was a large number of patients who had amyloid deposits in their aortic valve. So I'm sure it's there, but it's not studied.
So, how can we talk? Because, I mean, our participants in this movie, they are so deeply studied that I'm scared of even considering any extra examination on them. However, Partha, if I'm not mistaken, and I think, I'm not sure, I think David is on, but I think you guys are trying to develop some sort of measurement, or the only way of measuring amyloid in the heart is via PET-CT? Or are there other technologies that would be, let's say,
I mean, Dr. Bakari is the world guru on amyloid and the heart, and he will.
Is there any way for me to see Dr. Bakari? Because I see just the two of us.
Yeah, I mean, it's because he's in the there you
Anyway. Hello. Thank you. Thank you. After face to a name. Yeah, so maybe just so not to take the time of everyone else, maybe we should just meet and try to think together. Because I mean, think of what we're building now, and it sounds like you're gonna begin a cohort as well, and we could just join forces. I think it's a very interesting, and then again, I'm very excited about all this glycoproteomics, and as Partha suggested earlier, we could include their bloods on the batches that we send to the people at Weizmann to see if there is anything characterizing them that characterizes their amyloidogenesis in periphery and in the brain, in palm, right? It would be very interesting. So I think there is so much to do.
There's a huge potential in doing a lot of research with heart and brain, especially with amyloidosis, because there's so much there which no one has studied yet.
Yeah, and for all our fellows who are here, if you could just get a sense of the excitement between the investigators, and I almost feel like I should become a fellow again and investigate myself with all of you, because these are so brilliant questions and so amazing to be able to get all these tools to be able to do these kind of studies. I really mean it. And I know we are already 4 minutes. Anybody else has any points or question? I'm happy to bring here. But this, if you can understand, it's just the beginning of the breaking of the barriers of how we think, because a patient is one entity, and where multiple systems are involved, and we think about the barriers of the blood and the brain, and we think in an isolated, although at many times when we are making a decision about the care of the patient, it's the mother system, and often the brain may be the reason. Even if you want to do something, you may not be able to work on something. So I want to appreciate this time, Mikhail, for spending with us. And it's just the beginning of being able to be creative and be able to think outside the box and create new energy and enthusiasm and investigation that we can do and the potentials we have.
So I want to squeeze one more word. That now there are changes with the story with Fisher, but for the fellows, if you're there, please listen to me for a minute. And if nothing wrong goes on, we're supposed to get some nice funding to train fellows in dementia research and care. I mean, not in care, dementia research, no care. Care is a different story. And those will be opportunities for bringing people from different departments to work with us, and exactly what we said before, to take areas that are really tangent between the brain and heart in this case, and raise a fellow into a research realm. And also again, with a grain of salt because of this, because of what's happening now in Rutgers Health. But there is a major interest, and the chancellor, the vice chancellor, Bisher, in fact, is funding 2 years of fellowships for physicians who are interested in developing as scientists. So we have a big opportunity.
Fantastic. Thank you very much, everyone, for joining in. And I think we'll continue with the enthusiasm. Thank you, Michal,
Thank
for
you.
joining.
Thank you.
And I welcome our continued engagement and work together. With that, signing off. Good night. Bye-bye.